Hi, this is Dr. Jabali, and in this video we're going to focus on topic 9.4, which is all about plant reproduction. Here are some of the things that we're going to target. We're going to target about flowering and their gene expression. We're going to talk about how switching to flowering through the length of the light in the dark period. We are actually going to talk about pollination, fertilization, and seed dispersal. We're going to talk about how flowering has a mutualistic relationship with pollinators. We're also going to talk about the inducing of short days during out of season moments. We're also going to draw the internal structure of the seeds and draw, of course, the animal-pollinated flowers. As well as we're going to design the experiment to test hypotheses about factors that affect germination. So right now, before we can start anything, let's first learn how to draw the flower, since we're going to talk a lot about how the flower works. So in this picture, it is a perfect picture that you must know really well how to draw and be able to annotate and spot them. See, because IB expects you to know this drawing really well. So in one side here, you have the stamen, and the other side, you have the carpel. Basically, the stamen is everything related to the man, and the carpel has everything to do with the female. So when it comes to stamen, we have two sections, the anther and the filament. These are part of the male reproduction system, while the stigma, the style, and the ovary, they are part of the carpel, the female. Now you can see that ovary also goes with it. Now the sepal and the petal, these two are not related to the stamen or the carpel, but they are part in helping with the sexual reproduction of the flower. And we will see later on their jobs and their functions. But with this, we should be able to draw them very well and be able to label each one of these. So before I do, let me just get clear about what the sepal does. So the sepal protects the bud until it opens. We also have the petal that attracts the insect. We have the stamen that makes the pollen, and the carpels that grow into fruits which contain the seed. So this is the main general about each one of them. But now let's look and be able to draw. I usually start the drawing with drawing the girl first section, which is here the stigma, and then we have the style, and then we have a nice little circle to show the ovary, and then we kind of look inside the ovary with little circles like this to be the eggs, which we call the ovule. So I can label here as an ovule, which then I can say that this is the ovary, and this I can say that it's the style, and this is the stigma. So what I did here was just draw the female section. Now I'm going to use green to show you the male section, which here you can do the drawing, and you have the drawing here. Now this one here is basically the male section. This, of course, is the anthem, and this is the filament. Now these make up the man section. Now, of course, the man section we know as stamen, and you can see from the word man in there, right? And the blue section, all the that you see here, all of these ones, these are the carpel. That's the female section. Now, besides that, of course, there will be the petals, which goes surrounding that. And these petals here are just to attract, different types of animals to come to the flower. And besides that, we also have, let's use another color here. Let me use black. But it's usually, they have the petal that goes like this, and then he holds the plant. So that means the sepal. The sepal here basically is just protecting the plant. Oh, let me write the word for it, sepal here. So this basically is just holding the flower, protecting before it opens up. But it's only the stamen here and the carpel. These are the only ones that are actually the reproductive system. Now the petal here and the sepal, remember, these are not part of the reproductive organs, but they do play sometimes a role. Now, when we look at focus more on the anther here, the anther, if you look inside of it, the anther is made of like bunch of little pollens. And these little pollens here, each one of them, and I'm going to keep going, each one of them is actually a sperm. So we give the word for the sperm in flowers as pollen. So if I say pollen, I automatically know that's a sperm. So usually what happens is these pollens that are found inside the anther here, they have to leave the anther and land on the stigma, so which is sticky. So they stick there, and then these pollens just basically go down through the style and reach and join or fertilize with one of these ovules, and then that way it becomes a fruit. So that is the pathway. Now that motion, as you saw here, the motion of the pollen going towards the stigma and going all the way inside, that's called pollination. So we're going to see different ways of pollinating a flower. But before we go there, we have to know the two generations of of basically plants. Sometimes they can be considered as gametophyte, which that means they're haploid, and that means they're made of gametes. That means the sperm's an egg. And then we have the sporophyte generation, which is a diploid, and usually they produce spores. So what the plant does is alternate between these two generations. As you can see here, you have gametophytes, and one of those gametes fuse with the sperm and egg. They fuse and they become a zygote. Then they go to mitosis and they turn into a sporophyte. And then sporophytes goes through meiosis, and these spores now, they go through mitosis and they become gametophytes, and they keep going. Of course, reminded that spores is always a haploid, and gametophyte is always a haploid, and sporophyte is always a diploid. This is the maximum you need to know about these two generations. So sometimes plants work with gametes, and sometimes the plants work with spores. Now let's talk about pollination. When we talk about pollination, it's about flowering. It's growing a new flower. Now plants can use wind, insects, bats, birds, and even mammals. And what do? They transfer the pollen from the male section to the female section. So for example here, we have red flowers, they can attract birds. We get yellow, orange colors of the petal can attract the bees. And even the scent of the flower can make it easier to locate them at night. Why do we want them? Because they can help transfer the pollen from the anther to the stigma. So there's two ways of pollination. We have self-pollination and cross-pollination. So when it's self-pollination, there's not much variation. That means you're doing exactly the same copy of the flower. When it's cross-pollination, that means you're increasing variation. That means you're making mixtures of flowers, so you might have different types of flower. So here a little small drawing for you to get the sense. So let's say I have this flower here that is dark flower, and if I send its pollen to a white flower here, most likely I'm going to have a mixture, right? So you can see there was a variety here going on. And this motion going to the next flower, this was considered as cross. But if I have the same flower where the pollen of himself lands on its own ovary, and that means I'm going to create another flower just like it, then that will be self-pollination. So self-pollination means the own sperm on the own egg. Cross-pollination means the sperm of one flower moving to the next flower. So in the end, the whole fertilization basically starts with pollination, which goes from the pollen going to the stigma. And then, of course, the carpel will grow into fruits, which contains the seeds inside. So one example of pollination is the wind. So, for example, some plants like grasses, they do not have flowers, right? So they don't have the scalar petals and nectar to attract insects. But they do have stamens and carpels, right? They do have the female and the male reproductive system. So how could you pass on the stamens to the carpel? They use the wind. So if they don't have the petals, they can actually have the wind helping them. So imagine you have the stigma right here with the ovary here, and you have the anthem with the filament. So the wind will come blowing in, and that wind will hit here and cause one of the pollens to land on its own stigma and go down the style and cause the pollination. At the same time, this can also happen on the other way. It can happen as in this stamen right here of a stigma right here, and you have another flower right here with its stigma and ovary. So you hit the wind here, and then this thing just fly all the way and lands on another one. This would have been as a cross. So this is the self and this is the cross. So you can see how the wind can help with that. So fertilization. So the pollen grains germinate on the stigma, growing down the style to reach the oval, and thus fertilization happens. So the fertilization is happening in the ovary. Now, the fertilized ovule that I talked about inside the ovary, they will become the seeds, and the ovary becomes the fruit, so protects the seeds. So this is what you see right here. These are actually the protected seeds. They're basically coming from all of that part of the stigma with the style. So if you remember, here you had the stigma, the style, and then you had the ovary here with the ovules, right? So you can see that these little guys, tiny ones here that are highlighted here, they will become the seeds. And then the rest surrounding it here will be actually turned into the fruit. So the ovary becomes the fruit, and the fertilized ovule becomes the seed, and the ovary becomes the fruit. So now that we're talking about seeds, one of the requirements is that you must be able to draw a seed and know the functions of each part. So we already know the seed is inside the fruit, and we already know that the seed eventually is a fertilized ovule, right? So this seed has to be protected. Now, here are the parts of the seeds. We have the testa, which is a tough protected outer coat. We have the cotyledons, which is it's the seed leaves that functions as nutrient storage for the structure. We have the micropyle, which is a scar at the opening where the pollen tube enters the ovule. And we have the embryo root and shoot, which become the new plant where germination occurs. So these are the four sections of the seed part that you must know. Now, here is a drawing of a seed that you must be able to look at it, be able to annotate it, and be able to draw it. Now, some seeds, they actually live in the period of dormancy, which that means there is no growth or development until there's a right environmental factor that triggers it to germinate. So some seeds, they need fire to germinate. Some seeds need a good temperature to germinate. Every seed has a certain environment that it needs in order to germinate. And when we say germinate is when the seed rupture. So we have to know basically the drawing of the seed. So this is a seed that has been opened right in the middle. So you can see there's the micropyle right in the corner with the helium. And then you have the seed coat, which is the outside, and the seed coat usually known as testa. So that's the protective layer of the seed. And then inside, you're gonna find the embryonic root, right? And from there, you're also gonna find the embryonic shoot. So this shoot means it's gonna become the stem side, and this will become the root, obviously. And you have the cotyledons on each side, and of course, this is the entire seed, which eventually will turn into a plant. So you should be able to draw this. Now, examples of seed dormancy. So, for example, desert plants will only germinate after a significant rainfall. Many forest species require heat from fire to germinate. In areas with a harsh winter, the seeds require a long chilling period before germinating. So you can see that each seed has their own type of environment that allow them to germinate. Now, here are some of the seed's germination conditions in order for it to germinate. There are three things, and you need to know those three. The most important thing that you need in order to make the seed open and become a flower is water. Water is needed to rehydrate the dried seed's tissues. You need oxygen to allow the aerobic respiration to produce ATP, right? Because you're going to do a lot of motions inside the seed as you grow. And when you grow, you need energy in order to build. And of course, the right temperature. So some temperatures, depends on the seeds, they will have their own temperature, but each one needs to have a temperature simply because there's going to be a lot of enzyme actions, and enzymes work in a specific temperature. So again, once again, water, oxygen, temperature, these are three things needed for seed germination. So here we begin with the imbibition. The imbibition is the first part of the germination, which means just the absorption of water. It's when you drop that water on that seed. Now, because you drop the water on that seed, it will cause the seed coat to rupture, and this will start a bunch of metabolic reactions inside the embryo. So the first thing the embryo will do is release a plant hormone known as gibberellins. Gibberellins will stimulate the production of amylase, which is an enzyme. And this enzyme would start to digest the food storage of the endosperm, or should I say from the seed, the starch section, to turn eventually to maltose, and then maltose will turn into glucose. So if you ever had to write this in the test, when they ask you to talk about germination, so you have to write in the germination essay, here are the first step. The first step is imbibition, which basically means it's a drop of water into the seed, right? Now, what would that do? That would rupture the seed, and that will start the metabolic process. 3, that will cause the release of gibberellin. Now, what does gibberellins do? This is a hormone, and you got to mention that it's a hormone. This will do what? Activate enzyme. And this specifically in this is amylase. Then number 4, you're going to say that amylase eventually will break starch from the storage into maltose, and eventually maltose will turn into glucose. And why you need that? Because you need glucose to break glucose to get the energy needed to grow the plant. So this whole process of germination is just to get to glucose, because eventually using glucose for energy, because again, the plant is going to be built up, right? So that's the germination. Now, once again, where is the points that usually come? Imbibition, and then gibberellins and amylase, and of course the maltose to glucose, as well as starch, if you want to say here. These are the things that you will see come in the mark scheme, so they must be target. Now you have to mention imbibition is basically the drop of the water. You have to mention that. And you have to mention the gibberellin is a hormone, as well as amylase is an enzyme. And of course, why not say they doing all of this to get the glucose so you can start using the energy glucose to make the plant grow. So this is the process of germination that you must know how to write. So since we know about germination, and we already know that water must be available, right, for the imbibition, and oxygen must be required for the aerobic respiration during germination. What does that mean? Because here, if you remember from cell respiration, in order for you to break glucose, what do you need? You need oxygen in the mitochondria in order to get as much energy from glucose. So obviously you need oxygen here. And of course, we need the temperature, because once again, we have so many enzymes like amylase going there. So you have to have the right temperature for that enzyme to work. So remember these three. Now, these are three factors affecting germination. So if you're going to do a test of germination, once again, before I go to controlling flowers, you can play with these guys. So for example, you can design a test putting different amount of water and seeing when the seed does actually rupture, or you can have same seeds but putting different amount of oxygen and see how much the seed grows to see which one comes out used more of the energy. And then you can also have different seeds put in different temperatures and see which seed grows faster, and that means the enzymes work best in that temperature. So these are the three different ways that you can manipulate in a testing for germination. Now let's talk about controlling of flowering. When you talk about controlling flowering, it's called photoperiodism. What does the photoperiodism mean? The photo here is basically we're talking about the light that it's hitting. And periodism, basically it is the time. So it's the time that the light is hitting in. So the plant response to the light involves relative lengths of the day and night in the order to flower. So again, photoperiodism, it's all about the amount of, if there's light or darkness, and that will tell if the plant will flower or not. With that, we have three types of plants. We have the long day plants, the short day plants, and the day neutral plants. So when it's a long day, the flower when the days are longest and nights are shortest. When it's a short day, the flower in the spring, late summer, and autumn where the days are shorter, and flower without regard to the day of the length. And here are some examples for long days, such as radish, spinach. For the short days, we have all these ones, and then for day neutrals, we have all these ones. Now here is where you need to know for the IB. You need to know this chart really well. What exactly is a long day, what exactly is a short day, and a day neutral. Not only do you need to know them, but you also need to know examples. Make sure you know 2 examples for each. So out of this chart right here, pick 2 for the long day, pick 2 for the short day, and pick 2 for the day neutral. This way, if it comes an essay and you mention, you might get these points. Now, when I say a long day, what basically means it's a short night. So obviously says long day with the shortest night. Short day means you want a lot of nights, so that means you want the days to be very short, which usually is during the spring, late summer, or the autumn. So also you have to know the timing. Is it summer? Is it winter? When do they usually flower? So how does the mechanism of flowering actually works? It all starts with phytochromes. What are phytochromes? Phytochromes are blue-green pigments, and they are the ones who control the growing of the flowering plants, okay? So you have to remember that they are blue-green pigments. Now, there's 2 types of phytochromes. There is the inactive form, which we call the PR, and there's the active form, which is the PFR. So once again, phytochromes are pigments, and they control the flowering, and we have them when they're active or when they're not active. PR being inactive, PFR being active form. Now, usually we want to try to convert the inactive form to active form. That's because there's different types of wavelengths in there, right? So here's how it works. If you're in a red light, which is 660 nanometers, inactive PR is rapidly converted to PFR, okay? Now the active PR can absorb the far red light which is 730 nanometers. And in darkness, the PFR is very slowly converted to PR, basically. The slow conversion of PFR to PR helps the plant in time to night length. Now, this is a lot of information, but I'm going to put it in a way that makes your life much easier to understand this. But you do need to know, out of this whole process, what do you need to know? You do need to know, for instance, let's start with this, the type of light, if it's far red or red light. You also need to know who's turning to who. For example, the PR right here is going to turn to PFR, usually during what? During the day, right? And the PFR will turn into PR during the night. However, the PR to FR is very fast, and I should say somewhere here that it's very fast and say, but I'm going to show it. And the PFR, when it goes to PR, is very slow conversion. So this will affect the way the flower will do. So here we are going to repeat this many times to help you out. So we know the PR is the one that absorbs the red light, which is 660. And the PFR is the one that absorbs the far red light, which is 730. You have to know this. So what we see, you see the PR, here is the inactive PR. It wants to become PFR, which is active. So it has to be, the red light will be absorbed by this pigment, right? Because remember, these guys are pigments. So the PR absorbs the red light and turns into PFR. Now, the PFR is an active pigment and absorbs light, but it absorbs only the far red light, which only happens during the night. So during the night, the PFR is turned back to PR. So in this process, this is happening during the day, this is happening during the night, this is happening really fast, and this is happening really slow. Okay, so you can see that the day or the night will influence a lot on the flowering, because if you have a long day, you're going to produce a lot of PFR, agree? And if you have a short day, you're most likely to have more PR in there. So let's see how it works here. So like I said, here we had during the daylight, rapid conversion, PR, which is inactive here. I'm repeating this again to help you. And this is again is active. And what are they? They are pigment. This is a pigment. So this guy is going to do what? He's going to absorb during the day the red light and turn in a rapid conversion to PFR. And PFR during the night will, in a slow conversion, go back by absorbing the far red light and bring it back to this. Now the far red is a 330 and this one is 600 nanometers. You got to put the units. So I'm just repeating this because, again, you have to remember it as you go along. So to make your life easy, now when we talk about short day, when you see the word short day, what does that mean? A short day means you obviously going to have a long night, right? So in the short day, you're going to have the PFR, right, during the night, which is a long night. Think about it. So they're absorbing all the far red light, right? And it's going to be very slow during the night. So what happens? You build up a PR, right? Because remember, PFR is turned to PR during the night. Now, for the short day, that's exactly what it wants. It wants to have a lot of PR. And this way, it activates florigen, which is the gene that activates to make a flower. So the short day, what does it really want? The short day wants to have a lot of PR. So the more PR it has, the more flower it has. So short day actually wants what? A long night. Why? Because it just wants to turn the PFR into PR. So you want to make a lot of PR in order to flower. On the long day, again, think of a long day. That means this one wants what? A very short night. So the long day, what are we doing during the day? PR is converted to PFR, which builds really fast during the sunlight. And that means the long day says, oh, I like a lot of PFR, so I will activate the gene florigen that activates for flowering, and flower will happen. So what does the long day want? The long day wants a lot of PFR in order to flower. So if you want a lot of this, what do you want? You want to have a very short night because you do not want to go back to PR. You want to stick to PFR. So that's why you have a long day with a lot of conversion to PFR. So During the night, you barely going back to PR. You keep them as PFR, and this way the flower will happen. But that's for the long day plants. So here is a perfect, let's put a summary of this. So you have the short day. Now the short day plants, it's more if you think about the night, because that's the most critical thing. Short day just means that they want a long night. If they want a long night, what does that mean? That means that they want to turn everything into keeping it to PAR, right? So let me erase, no, actually keep it here. They want to have large amount. So large amount of PR. So that means they want all the PFR to be converted to PR during the night. That's why it's the long night. If it's a long day, if it's a long day plant, what would it do? That means they want a short night. Remember, the critical thing is the night here. So with that, what do you want? You want to have PFR large amount. And since you want a large amount, you want in a short night to have not much of the PFR. You want most of the PR to be converted to PFR during the day. So this way you can get the flower to happen. So if you remember these two, good. Now, usually I tell students, you can see the word short, and you can short for the R, you can see the R is a little tiny. And then when you think of long, think of PFR, which is the F here is long. So long always wants PFR in a large amount, and short always wants the PR in a large amount. This is the only way they will flower. So making a quick summary again, this is after you can stop this and copy, and then I will erase this and make it even more straightforward. Just think of this way. Short wants a lot of, in this case, PR, and the long always wants a lot of PFR to flower. That's straight up. And what's the most critical thing here is the night. It all depends on the night. Many people think it's the light, but it's actually the night. The night is what defines if you're going to flower or not. So plant flowers only after critical day length. Plants flower only after critical night length. But the day length is not as critical as the night. So the night is the most important thing. So the multiple choice, remember, the night is the most important thing for the flower. Now we have examples of flowers that you can see how they flower during the day or the night, depending on what kind of plant they are. Now here's to show you why night is the most critical thing. And this is a picture come from BioNinja, which is perfect because it will literally show. So if you remember the short day plants, they need a lot of PR, right? So as a PR, they want to make sure that in this case, that they don't have much PFR being produced, right? Because when does the PFR produce? During the day. That's why you don't want the conversion to happen. See, in a long day like this, you see this is a long day, what happens? You end up making a lot of the PFR turn to PFR, which is a no-no, so no flower. Here, the short day, right? And then you have a long night. That was great. Why? Because in the long night, you turn the PFR to PR, and great, so you do flower. Here, you do have a short day. However, in the middle of the night, you stop the production of PFR to PR for a while. And what happens? That means you end up having less of PR, and that means no flower will happen. So one simple light bulb turn on inside, let's say, a greenhouse can stop the entire flowering of the plant. So when a short day is a short day, it really means it wants a long night completely dark. When it's a long day, that means they don't really care much as long as they get a long day. So here in a long day, what's happening? You're going from much of PR to PFR, which is great, flower. Here, if you're not getting enough of the day, that means not all PR will produce PFR, and here you will have most of the PFR turn into PR, so no flower. Here, you did produce most of them to PFR, and then you have the extra amount here produced, so flower. So it's the night that should be critical. If something goes wrong during the night, it can affect the whole process. So reviewing again, short day plants, the PFR inhibits the flowering. Long day, the PFR activates the flowering. So here, what do we want? We want the PR. This one, we want the PFR. So PFR levels must be lower for the flower. And they must exceed the critical night length. This is so PFR must be higher to flower, must be less than, the night might be less than the critical length. So in the end, short wants a lot of PR and long day wants a lot of PFR. And that's pretty much it for this topic here on 9.4. These are the study guide questions. You can easily answer these questions. It's found in the video. And if you know how to answer them, then you have mastered 9.4, which is the plant reproduction system. And that's pretty much it. Thank you.